The 2009 Lasker DeBakey Clinical Medical Research Award honors Brian Druker, Nicholas Lydon, and Charles Sawyers for groundbreaking work on the treatment for chronic myeloid leukemia.
I was told by my then oncologist my only way of dealing with CML was to have obviously the highest forms of chemo and radiation possible, and then to have a bone marrow transplant. You know, I didn't think my children would grow up knowing me.
Dr. Brian Druker, an oncologist dismayed by patients dying, was convinced it was possible to develop a drug that would specifically attack the molecule responsible for causing the fatal disease CML, chronic myeloid leukemia.
But there was no proof of this concept. And the field was waiting, just waiting and waiting for this proof of a concept. It hadn't been done before. Good girl.
In the mid-80s, the young biochemist Dr. Nicholas Lydon becomes interested in the emerging data on the pathogenesis of CML, where one particular kinase enzyme goes awry. Lydon is fascinated by the prospect of creating a molecularly targeted drug.
So the challenge became, in a very short time, quite daunting, how to get a selective compound that would address this defective oncogene without having the same type of drug that existed before, a generally cytotoxic drug which inhibited all kinases.
And Nick's group at DeBakey was one of the few in the drug industry, pharmaceutical industry, that were interested in developing inhibitors of this enzyme family.
This is the shortened Philadelphia chromosome, shown here in diagrammatic form. My vision based on the pathogenetics of CML was that if you came up with a small molecule drug that would specifically bind to this protein and not to the other proteins in the body that use the same mechanism, kinases, that you would be able to address the disease. And that was quite a challenge at that time, because most people felt you really wanted to have a cytotoxic drug. They were the only ones that worked. So our view was you could potentially use these targeted therapeutics.
Most other large drug companies looked at this family of enzymes and said there were all sorts of reasons why inhibitors of this family will never work, and they wouldn't touch it.
When Dr. Druker sets up his own lab in 1993, he's totally focused on CML and immediately contacts Dr. Lydon.
And he reestablished contact with me in around about 93, I think it was, when we sort of identified imatinib. We hadn't fully profiled it. We weren't convinced that was the final drug, but it looked very promising at that step. And he was very interested in CML, and he was looking for companies who had inhibitors, because that was his vision. He wanted to develop a drug that would help patients, obviously patients he was seeing in his leukemia clinic. At
that point, he had developed inhibitors of this enzyme, and he was willing to let me test him. So within 6 weeks of my arriving here in Portland, Oregon, I had compounds that inhibited this enzyme to test in my laboratory. And one of them stood out head and shoulders above all the other compounds. It unfortunately wasn't their lead compound, but I was able to provide data to Nick that allowed him to put resources into a project.
I think it convinced DeBakey this was a good target. I think if I hadn't had his assistance, I wouldn't have had the credibility to persuade them this is going to work, or this should work.
I redoubled my effort to get this compound into clinical trials, and quickly. They need something now. They can't afford to wait.
Our expectations were not that to get these amazing results. We were sort of a little bit more tempered in our expectations. But it was an extremely exciting time for me.
I knew that we had something that we had never seen before.
That was the first time I thought, wow, this could work. Before that, the project had its ups and downs, and we were holding our breath.
Dr. Charles Sawyers and Dr. Druker, in cooperation with DeBakey, designed and implemented the clinical trials. Everyone is amazed at the results.
One of my patients was, I believe, the first patient to have what's called a complete cytogenetic response, when her bone marrow test came back as 0 detectable Philadelphia positive cells.
Much to everyone's dismay, patients in the late stage suddenly start to relapse.
Some of the mutations cause Gleevec resistance, lock the activation loop on. And that's where Charles came in and very quickly understood that it was mutations, changes in the BCR-ABL kinase. The target of this drug had shifted, and the drug could no longer bind, no longer be shut down. So we learned what we already knew about cancer, is we had a formidable enemy, that it had figured out a way to evade our therapy.
We started seeing this pattern of relapse develop in the blast crisis patients. We began wondering, can we understand what's changing in these patients' tumor cells that's causing the resistance to develop? Why isn't the drug still blocking the enzyme? It's blocking it, last week, but not this week. So the upshot of that is we discovered that most of these patients had new mutations.
Within a very short period of time, a new compound, dasatinib, is identified, which inhibits the mutated form of the enzyme.
So by having the three-dimensional structure, a picture of how this drug binds to the enzyme allowed drug companies very quickly to circumvent resistance mechanisms.
The journey to discover a CML treatment has brought the promise of life to both doctors and patients.
I believe that will continue to be not only my miracle, but open many doors. And I've enjoyed my life tremendously over the past 8 years.
And the patients I see have chronic myeloid leukemia, and they're treated with a drug that I helped develop, and they're living active, productive lives. And I get to see that with my very own eyes.
They're looking at me for, in desperation for help with what they knew was a terminal disease, and to realize, not only how brave they were to go on this experimental study and then to benefit and then to share in the excitement and the discovery all along the way was, it was clearly a life-altering experience.
It's working in patients, that's something I guess that everybody who works in drug discovery, dreams of, that they actually do something that ends up being useful.
Today, CML is no longer fatal. Many scientists and clinicians contributed to the development of the successful molecularly targeted treatments. But because of 3 people, it has become a reality through the work of a lifetime.